Hello everyone. So today we're going to talk about transcription and translation. So we talked about replication, which is making a copy of DNA in order to make new cells. But now we're going to talk about transcription, where we're transcribing DNA into an RNA, and then we are translating that RNA into proteins. So the whole purpose of this is to create proteins. Okay, so we start with DNA, we go to RNA, and then to proteins. So that's how it all starts. DNA to RNA to protein. Okay, it's a whole process. So as you can see here in this image, it's like a cell. Inside we have the nucleus. We have our DNA, and inside the nucleus we're going to transfer that to an RNA. Why an RNA? RNA has a base that's a little different, and it has a different composition or structure, and it behaves differently. So RNA can be mRNA, which is a messenger RNA. We can have a tRNA, which is a transfer RNA. We can have an rRNA, which is a ribosomal RNA. So the RNA kind of shapes itself to help out with the process of making proteins. Okay, so there's different types of RNAs that we're going to make, but it's very important in order to make a protein. Also, the proteins, the ribosomes, everything's going to be outside the nucleus. We don't want our DNA to exit the nucleus. We don't want to damage our original DNA. So right here we're talking about a certain region or a gene, certain area that codes for a protein. So we'll say this little section here is going to be transcribed to RNA. That little section is going to leave for the proteins to read it and create another protein. This is ribosomes. To read it and create a protein that's going to be used all over our body. Okay, so we don't really want the original DNA to exit. We just want like a copy or an RNA or messenger RNA to send the message out and create the proteins. Okay, and so DNA has the basis, if you remember, A, I'm just going to abbreviate, T, C, and G, while RNA has the basis A, U, C, and G. Okay, and that's the biggest difference. It's just one base that's going to be a little different, the composition, so it's a different letter that we're going to replace, and that's our RNA. Okay, so we'll go with the process of transcription very quickly. So this is kind of what it looks like. I like this image a lot. Okay, so think about the DNA, think about all the little bases that are in between. So this is our DNA. So through transcription, we're going to open this up a little bit, and then we're going to create an RNA copy from one of the strands. Okay, from the template strand, we're going to create a copy. Now that copy is going to leave, which is our transcript or our RNA, and then we're going to translate all those letters into amino acids, which will then make a polypeptide chain or a protein. Okay, so that's the whole purpose. So this is transcription and this is translation. It really is simple, but sometimes we complicate ourselves a lot. Okay, so we'll start with transcription. Transcription. Oh man, that's horrible. Right. Leave some space in here. Underline this. Basically, they're all defined by, or they're all categorized by three different steps. We have initiation, we have elongation, and then we have termination. Okay, so those are just the three steps. So where it starts, where it elongates, and where it finishes. Okay, initiation. So you start with a promoter or a promoter region. So remember, this is a part of the DNA. So it's like a gene, say one of the genes to create a protein. So those parts of DNA have a promoter region. That promoter region is where it promotes the beginning of transcription. Or the RNA polymerase is going to come into that promoter, and that's where it's going to start. So the DNA is going to unwind, kind of like open up, kind of like in replication. Okay, RNA polymerase attaches. Polymerase attaches to the DNA strand. Okay, DNA unwinds by helicase, the other one, and then That's basically the initiation. That's all it is. So you start with the promoter, RNA comes in, and DNA starts unwinding. Okay, so we start opening it up. Elongation is where RNA polymerase begins adding bases to the template strand. Remember how in replication we would add the letters as well? But see the difference here? Instead of adding a T, where it's A with T and C with G, we're adding a U. Okay, so that's a little difference. That's a big difference actually. Okay, so the RNA, this is going to be our copy. This is our original on the bottom. This image doesn't have the other one that's on top. So think of this, the original, the bottom half. And this is a copy of RNA. Okay, so basically adding bases all throughout to copy the whole gene sequence, or the whole sequence of letters that we have. Okay, polymerase, I forgot to add an E. The template strand. Okay, so this is kind of what it looks like in, I don't know, maybe a more detailed picture. We have our DNA down here. This whole thing's a ribosome, like a guitar pick, and it starts adding the nucleotides, the RNA nucleotides, base by base by base by base. That's basically elongation. Okay, it's really simple. It's just adding the bases and making the chain of RNA. And termination. There is just how there was a promoter region, there's a terminator region. Okay, and that's where it tells RNA polymerase to stop. It's basically what it is. So you terminate, or you end up with, you can have an mRNA, a tRNA, or an rRNA, depending what the function is and what the purpose is. And I'll show you the differences in a little bit, okay? But that's basically what we end up with in transcription. A copy of the DNA in the RNA form. So now that copy is going to exit the nucleus and go into translation. Okay. So now again, we had DNA, we made the RNA. Now we're ready to translate it into a protein. Okay. So now same thing. Now let's go into translation. It's very similar. It's very similar in the 3 steps that what they're called, but the process is a little different. Okay. So now we got translation. Prefer to pause it if you think it's a little confusing. But okay, so say this is our RNA strand. Okay, so this is a copy that we made. Each letter is going to be down here, each base. So the way this works is that every 3 bases create a specific amino acid. Okay, and that's how we have a chart here. Let's see if I can show you the chart. Here's our chart. So every time you go to 3 letters, you get a specific amino acid. Okay, you could get valine, alanine, threonine, proline, serine, cysteine, arginine, histine. Like there's so many, lysine over here. We have amino acids that are specific to the 3 letters in the RNA. So what's going to happen? We have the 3 letters like AUG. So we go AUG and find it over here. Where is it at? AUG. Maybe you found it before me, so you go with the A. So the first one, A, the U, and the G. The MET, or methionine to start. So this is our start codon. So these are called codons. Every 3 letters, that's a codon. We always start with a start codon, and we end with a stop codon. Okay, so you start with a start codon, which is AUG, and then you add all these different amino acids, letters and letters and letters and letters, and then you end up with a stop codon. So in this example, we have UAG. Okay, so UAG right here, stop codon. Okay, and that's kind of how we create those chains of amino acids to then create a protein. All right. So initiation, same thing. Initiation. tRNA. There's something called tRNA. Binds to the mRNA. Starts reading the codons. Sorry, the ribosomes. sorry, this is the ribosome, my bad. I got ahead of myself. The ribosome binds to the mRNA and starts reading the codons. Okay, so here's, I think this is a better picture. So this is basically how it goes. Okay, so this big, it looks like a snail, this big green circle, that's what's going to be the ribosome, and it's just going to jump in there into the RNA and start reading. Okay, these are tRNAs. Let me see if I can draw. So let me try to show you first, and then we can write it down. Okay, so these are let me zoom in a little bit. So these little things, these are our tRNAs. Okay, so this is our codon region right here, and this region, these 3 letters, is called anticodon. Okay, so this is our anticodon. What does that mean? It's just the opposite of the codon. So every tRNA has a specific anticodon and a specific amino acid. So this ribosome being attached in here is going to call for that tRNA to come in and attach to its section and leave an amino acid here. Okay, once it comes in, another one is going to come into the next 3 letters, attach itself while kicking this one out. Okay, so here's our first step right here. The tRNA came in, it's in the middle. They're called EPA sites, or different sites, just where they exit and when they enter and when they're extending in the middle. Okay, so tRNA is coming in to the 3 letters. It attaches, and then the 2 amino acids, or say that for right here we're on the 3rd one, each one of these amino acids. So the first 2 that were already attached are going to attach to the next one. So now you're going to have 3 in the middle, right? So see on this picture on the left, now you have 3 of them. Boot this one out. This one jumps to the middle, and then a new one's going to come in, and then add to those 3 and make it 4. Kick it out, move to the middle, and then a new one comes in, and that's our whole cycle. It's over here. Kick it out, bring it to the middle, and then another one's going to come in, and then keep going and going and going and going and adding the amino acid sequence, creating a big protein. Okay, that's a whole purpose. So that's all elongation, basically, where the tRNA is just going to come in and start adding, start reading the codons and adding the amino acids to it. This is kind of what it's going to look like, a little more visual, or a little more in detail. Okay, so you have your codons on the bottom. You see the letters. So this one's more specific to the letters. These are your anticodons over here. So this is your tRNA. Remember how we made mRNA? We can make tRNA. That's going to help it out as well. So this is one of the tRNAs. These are the guys over here that just have tRNA with its amino acid. tRNA with its amino acid over here. So they're going to come in, and they're going to attach inside, bring in that amino acid, and then just keep making this long chain or polypeptide chain of amino acids to create protein. Until when? When do we stop? Until we reach a stop codon. Okay? So we're going to keep adding and adding until we reach a stop codon, and that's where we're going to stop reading the mRNA, and that's where the protein is created. So let's write down a couple things, okay? Hopefully you didn't forget. Okay, so initiation, so just the ribosome is going to bind to the mRNA, start reading codons. Elongation, tRNA, tRNA reads the codon and attaches its anticodon, leaving the amino acid and attaching it to the chain. All right. So it's just going to keep going and going. So that's what we had here in this picture, in these 2 pictures. tRNA is just going to come in, read the codon, attach the anticodon to it, and then just leave its amino acid in there and be kicked out. And then at the end, on termination, stop codon signals the end. Polypeptide chain is created, or a protein. Okay, then we create a protein at the end. And see if I have an image here. Yep, so here's our big chain that we're gonna have at the end. Well, that's only 3, right? But there's a big one. Yeah, so this is basically the whole process. It's a cool picture. I'm gonna send you these images, okay? With the DNA replication. So basically in here, inside, you can see, let me see if I can get a highlighter. Here's our RNA being read, okay? It could be RNA, mRNA, tRNA, whatever. Then it's gonna exit the nucleus. It's gonna be outside. The ribosomes or the ribosomal units are gonna attach to it and then start adding the tRNAs inside, creating this long chain, and you end up with a protein. And that's translation over here, and that's transcription inside the nucleus. That's translation outside the nucleus, okay? It's basically transcription and translation. It's just adding bases and creating reading. So creating a copy of the DNA to RNA, leaving the nucleus, reading it, and creating a protein. Okay, now it's very important, very specific. Why? Because think about this one right here. You have GGC would make to stop drawing. There it is. Okay. Here we have the different sequences: AUG, AAG, UUU, GGC. But if you change one letter right here, instead of GGC, we put AGC. It changes the amino acid from glycine to serine. What happens is that change of amino acid alters, remember, the folding of the protein itself. So if it alters the folding, it alters the function of the protein. So when you have a wrong base or a wrong letter, then you have a wrong amino acid. If you have a wrong amino acid, then you have a wrong protein or protein that doesn't work. So now you're coding and reading proteins and creating proteins that are not going to work. Okay, so it's very important, just one single base that can create chaos inside our body or inside the cells. Okay, so that's the importance of that. Different mutations, right? And I'll see you all on Wednesday in class.